Hello guys, how are you? My name is Tatiana Petrisky and I'm doing my presentation for Emory University for MI390R, and my topic is the neurobiology of addiction, the perspective from MRI, present and future. And I found this topic to be very interesting, and I just want to share my knowledge with you. Okay, let's talk about that. So, addiction research has been benefited greatly from neuroimaging. Using radiological examination to understand the consequences of chronic consumption of alcohol has been an interest since 1950s. MRI is the primary choice for addiction imaging research due to its safety. New methods and technologies are examined for future opportunities to advance knowledge of the neurobiology of addiction. So what kind of methods are we using? We're using voxel-based morphometry. VBM is the most prevalent methods for measuring brain structures with the visible contrast between white and gray matter. High-resolution MRI helps to estimate tissue volumes together with ventricular volumes filled with cerebrospinal fluid. Reduction in cortical volumes are noticed, especially in the frontal lobes, with almost all cases of drug abusers. Subcortical structures associated with dependency on stimulants such as cocaine, meth, increase in volume. I found this picture and I found it very interesting. So just like a basic knowledge how the voxel-based morphometry is done, and so you get the original image and then the image is normalized and the gray matter segments are constructed. It's right here, the third from your right, from your left, modulated GA, gray matter, and smooth gray matter. So yes, I am looking forward to do my MRI so I can learn more and more about, but it's going to be next year. So now let's continue. So what kind of methods we're using? Different addiction groups had shown reduction in gray matter volume compared to healthy controls. In regions of the brain targeted by a specific drug of abuse, including alcohol, cocaine, and amphetamine and nicotine. White matter differences have been difficult to observe with whole brain VBM. Detection of these changes are associated with amphetamine use. The other method we can use to detect changes, it's called diffusion tensor imaging, DTI, an MRI technique of imaging the diffusion of water and estimating the trajectories of white tracts. DTI produces maps of the human connectome, which can help to overview the measurable DTI effects in chronic alcoholics. A reduction of white matter in anterior parts of the brain, particularly the supratentorial and infratentorial white matter bundles with sufficient myelin degradation. Functional MRI. This is the picture I found, and I found it very interesting. This is the pre-treatment after 4 treatment sessions. Functional magnetic resonance image indicates significantly less craving-related activation in the brain after treatment. The different colors highlight its changes between pre and post, but have no meaning beyond that. This is functional MRI, measures the brain activity in the blood oxygenated level dependent contrast bold. MRI detects magnetic properties of oxygenated and deoxygenated blood. Experiments with functional MRI often induce cognitive processes with appropriate stimuli, highlighting the regions and circuits that are involved. Emotional response is activated the limbic system in alcoholics, primarily in insula, inferior frontal, medial temporal, and parahippocampal regions. parietal and medial prefrontal regions of the brain show increased activation in cocaine addicts. Surface models of the cortex. Representation of great matter covering the cerebral hemispheres can form from the inner, gray-white, or outer pale surfaces. Measures of the cortical thickness, volume, and geometry are complementary to the volume estimated from the VBM, which is a combination of thickness, surface area, and gyrification. Thinner cortices are seen in dorsal, prefrontal, and insular regions for cocaine addicts. Alcohol shows a decrease in cortical thickness to frontal, pre- and post-central temporal, and occipital cortical regions. The other method we can discuss pharmacological MRI. Pharmacological MRI is used to test how pharmacological agents modulate brain activation or cognitive performance during pharmacological MRI study. The use of pharmacological MRI may allow neuroscience research of addiction to make more informed assumptions regarding the neurochemistry of underlying disturbances to particular cognitive processes in addiction. Susceptibility-weighted venography. Cardiovascular illness is a major risk factor for addicts. It may lead to increased risk of intracellular hemorrhaging due to changes in cerebral flow detectable by imaging. MRI field strength is capable of detecting small structures, vessels, less than 1 millimeter, relevant to addiction research in areas of the thalamus and midbrain. In the conclusion, I would say that MRI imaging is the only one part of wider effort to understand the etiology and treatment for addiction. New techniques will lead to new opportunities to understand or mitigate the impact of addiction. The monitoring of abstinence and prediction of relapse are key to supporting clinical decision making and would be a significant step forward. And this is my reference list. And then have a healthy lifestyle, guys. And I just wanted to say Merry Christmas and Happy Holidays to you, and I will see you soon. Bye.